Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our final talk, Mapping the Human Body at Single-Cell Resolution, Developing the Human Reference Atlas. I'm incredibly pleased to welcome our final speaker, Supriya Bidanta. Supriya is a driven and dedicated research assistant at Indiana University, currently affiliated with Katie Borner's lab. Her academic focus is on bioengineering, which is complemented by a minor in bioinformatics. During her talk, Supriya will discuss insights into the functionality of individual cell types and anatomical structures with reference to data gathered at single-cell resolution from various organ tissues. A very warm welcome to you, Supriya. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I would like to hand over to Supriya to begin her presentation.
Hello, everyone. I'm Supriya Bidanta. I'm a part of Katie Borner's lab here at Indiana University. So the topic of the talk is mapping the human body at single-cell resolution, developing the human reference atlas. As you know, having an atlas would help everyone to know where to navigate to and give the key insights about how everything is there as an overview. So here as a lab or as a project, we do the same. Moving forward. Oh, okay. So what is HubMap? HubMap is the Human Biomolecular Atlas program where we map the human cell anatomical structure and the vasculature into a virtual human being where we can just see the integrity of the human body. This project is a computable open source map of the human body at single cell resolution. It is funded by it's funded components with 350 researchers in more than 50 research units across US and Europe. The initiative for HubMap was to build a tissue map with the possibility of visualizing, searching, and modeling data with help of infrastructure, engagement, and communications. Here at Indiana University, as a part of Katie Borner's lab, what we do is visualize we store the data and validate the data and so forth. The rapid technology implementation, what we are doing is to enhance the large-scale validation, integration of emerging new technologies. The center as I said, the center collects the data and analyze a range of normal tissues across multiple organs. The transformative technology development we do is analyze, like for visualization, validation of new methods for mapping the body at high resolution. The project as a human biomolecular atlas program is also collaborating with multiple different institutions like KPMP, where they majorly work on kidney, human cell atlas, Select gene, where we have a lot of huge data. And we collaborate with Allen Institute for brain science and JetX for the skin data. And there is human protein atlas as well. As I mentioned before, HubMap as a part of here at Indiana University, our major work is on integration and visualization engagement. That is a part of like data storage, dissemination access, and map generation, where map generation is like virtually creating organs, tissues, and mapping it to the exact location in the human body. As you can see from this slide, the tissue mapping centers are across the US, where it varies from like west to east, with California, Washington, Texas. We have centers all over, all across US. Having said that, we have like different organs. We have donors from different organs, be it male, female, with different modalities like mass spectrometry, microscopy, and sequences. Coming to the main question is like, why do we need a human atlas, and what is the point of a single cell? As I mentioned before, human is a complicated creature in this planet, and it's very important to know the integrity and know the complexities of the human structure, and it's very important to have the healthy have the information of a healthy body. So human cell atlas does the same achieve the same goal by multi-scale, multi-dimensional mapping of the human body for the whole body level to the individual cell, coming from like head to toe, from like brain to muscles to like bone. What kind of cell structures are there, what is their functionalities, and how is it important, and what is the minute level, like what is the exact positioning of those cell, and the biomolecular that are used to identify. These are the things that we really focus on as a team. Is it possible with this project? It is possible to classify cells by their expression profiles, as we are advancing technology from like sequencing, from snRNA sequencing, then attack sequencing, then snRNA sequencing. It becomes easier to locate and know the functionalities of each cell and the vasculature that is there in the human body. Considering the human cell data, we know that it's a large scale data set, and it is very important to have a machine learning tool that will help us to predict and visualize the tools to find how cells are related to each other and how they interact with each other. This is really important to have an have an cell information about how the cell interaction happens within the body. Again, this project enables the creation of cellular references maps of the position, functioning, and characteristics of every cell type in the human body. The challenges that we face as a team is like how to know that the positioning, the exact positioning of the tissue in the human body. It's very easy to see when we dissect the body or like see it how it is there, but it's very difficult to virtually map it every cell or like every organ or every tissue unit that is there in the body. So another part of the challenges that we face as a team is like, what are the cell types that are there, what are the functional and what are the important cell types within the anatomical structure with various filters in place. For example, like in a live tissue, there will be like blood, there will be different lymphocytes, there will be like different things that are not visible, that is not required or not required. I mean, it blocks the view of it blocks the exact view of the human body. So that is where it becomes difficult to see exactly what kind of a cell, what kind of a what is the positioning, and yeah. So the first thing that we do to understand the human body is to map the tissue into a registration into user interface, where we create a 3D model of like 3D model of like the section of the tissue that is there. For example, in this slide, we are showing the part of a kidney, where the tissue block has an orientation where we can move with X, Y, Z. We can orient the block of tissue, like what is the exactly positioning? Is it like aligning with the tissue that is present in the whole tissue that is there in the image? Coming to the next, we have exploration user interface, where once the registration has been done for multiple tissues, we have an interface where we can explore the data. Exactly, is it the perfect match, or is it perfectly aligning with the tissue block which we have in our database? So as you can see on the left hand side scrolling, that we have heart, kidney, with multiple subunits of like papilla, kidney capsules, renal medulla with renal pyramid is the one which is showing on the right hand side. And there are renal cortex. And so for now, we have mapped a lot of organs in our database, and this is just one organ that I'm showing here right now. Okay, once we are done with the exploratory user interface, we come with the organ reference, or 3D reference organ, where we can just replicate the data which we have in our database to a virtually or a 3D structure, where we can just view the organ as it is. It's there in the human body by mapping everything, and with exact color of the body, and like exact color of the tissue and everything into a virtual 3D model. The open source library of 3D models derived from the visible human data set. You can go to this site and look at it. It's really cool. And first, initially, the 3D organ was funded from 2016 to 19 by National Library of Medicines. Then it was moved to NIAID in 2009. Now, from like January 2022, this is a part of HubMap Hive structure. Yeah. Now, the challenges that we face while modeling a 3D reference organ is like, can we replicate the exact textbook appearance? To be honest, it's like, it's more of like we question the textbook thing. Is it the exact thing that is present in the world, exact thing that is present in the human body? So it's kind of difficult to adapt the real data to real data of textbook into appearance. Then the anatomical subdivision dictated by ontology, overlapping jigsaw pieces, carving up original model. So while modeling the 3D organ, as you know, the human body is so complicated, so convoluted, it becomes difficult to map every minute detail of the human body. So the anatomical subdivision dictated by the ontology is like overlapping structure is kind of difficult to map in 3D organs. High degree of variation in resolution between or within the model. As we are mapping in the single cell resolution, it's very important to have a fine defining structure of the cell type that is there. And when going to that level, of that minute level, it becomes very difficult to the resolution disrupts the resolution becomes very hazy, and it becomes very not so clear to the human eye, which is like a difficult to understand the human atlas as such. The third part, the next part of the difficulties that we face is creating and placing organs in no particular order without surrounding structures to help orient. As you know, like we are human, as a human, we have like different organs which are like interconnected, and to build a piece, it's like a Lego. You need to have that kind of a you have all the pieces, but it is very difficult to know what is the actual order of each Lego pieces that you can use to construct the human body. In this slide, I'm just showing two pieces of one. On the right hand side is the small intestine measurements. As you can see that as we go by section and section, it becomes very difficult to like map everything into this convoluted, highly convoluted small intestine. And on the right hand side, on the left hand side here, I'm showing a breast model where the actual blue region is the skin and the gray is the adjusted one. So it is kind of the challenges that we are facing for as a project. For if you want to like view the whole set of organs that we have mapped, for now you can just scan through the QR code and you can visualize all the organs that are there. For now, we have blood vasculature that is 3D modeled. Then we have bone marrow, then placenta, prostate gland, uterus, and yeah, there are a lot of organs that we have mapped as of now. Coming to the real data set which we use, that is the anatomical structure, cell type, and biomarkers. It is the ACTB table, generally we refer to, which has the organs, the anatomical structure, and the cell types and the biomarkers. This data set is in a CSV and is publicly available. You can go to the link that is there underneath and access the data. From this portal, we can do the visualization, how everything is linked from like the organ with edges and the nodes that is there. And the data from this assay analysis indicates which anatomical structures, once all the data has been processed by from the assays, we store it in the ACTB table. This data set we use to know the functional tissue unit of each organ, like and the vasculature that is there to map each FTUs of the organ. This is a sample example of ACTB table that I'm showing right here. It's a kidney. As you can see that we have like various columns. The A stands for the anatomical structure. The CT stands for the cell type that is there. And going forward to this sheet, as I couldn't fix it into this slide, we have biomarkers as well, and the reference file which from where we are getting those data. Along with the anatomical structure, we map it with the ontology ID and the Uberon IDs for it. And for now, we have like for the ACTB table, we have like over 25,000, more than 25,000 nodes and edges as a part of the knowledge graph. If you want to explore the data, we can just you can just navigate to reporter page and view all the data sets we have. We have like over 32 or 35 organs for now. Going forward, as we talked about 3D organs, we have another project that is called the 2D functional units. 2D functional unit is a cross-sectional of the organs which we have till now. For now, we have mapped almost like 10 organs with 22 FTUs. Functional tissue unit, I would say, is the tissue unit that is really important and plays a functionality as an important functionality to the organ. Like for example, we have mapped renal corpuscle, which is which is an important part of the nephron. And as you can see, we have like mapped from like 0.1 millimeter to like 1 millimeter of sizes of FTUs. We recently published a paper on this you can go to this link. This is a preprint though. You can go to this link and read through the paper and let me know how it is. Coming back to the point. So for now, we have included kidney, large intestine, small intestine, and spleen as a part of like FTU illustrations. This part of the process is done at a team of two, where the medical illustrator designs all this, and the medical illustrator is provided with all the information from the ACTP table which we have, along with it data curated from the literature database that we have on the internet. So we try to detail every minute and every exact detail of the FTU. We coming forward, so all the data, all the information that I have talked about till now can be found in the Human Reference Atlas portal. Here is the QR code, and you can just scan and look through it. It's a really pretty portal, and give a lot of information on what are the data set we use, like coming from the codex data, or like the sequencing data, and how do we visualize it, and what are the mappings which we do. Coming forward to it, as we have mapped everything now, so there is a visual resource, visual resource over here, where we can just wear the goggles and just navigate virtually and how it works. So for this part, I don't have much information, but if you want to know about how the organ gallery works, you can contact Andreas Buckle. He is a really great person, and he has a lot of information, and he is really good at it. So the next part is visual resource, where we see the the cell type, tissue type, the actual tissue type that we get from the donors, and what are the cell types that is there. So this is a part of our project where we do the cell segmentation on the actual data set, actual tissue slides, and then map it with the cell type and the immune cells that is there. This is a part of the example, what I was talking about, as a part of vascular common coordinate framework. Here, as you can see, that we have a tissue block, and in this tissue block, we are trying to map the vessel cells, and the distance from the kind of immune cells that are there, and how far is it exactly from the endothelial cell. From this graph, from this plot on the right hand side, we have the tissue unit, and for each we have like multiple of like 24 slides, on which we section it accordingly, and we get the information about the damage that is there, the cell types that are present in the tissue, and what is it, the immune cells that are there, and how far is it from the endothelial cell. As a part of this project, we also we are working on skin, mostly skin and large intestine. As of now, we have information about that. So coming to the final slides, all the data which we have for now, all are like publicly available, and it's easy to access. You just have to Google HubMap, and then you'll get the information about it. We have a short tutorial about how to view the user registration user interface, then exploratory user interface, and 3D organs and 2D FTUs into this in this tutorial. Coming part to the data portal, we have like a visualization tool, as I mentioned before. I'll quickly show you how does it look like. We have a page called the ACTB reporter, and we need to click on the go to visualization, and we have the ACTB table. For now, I'll just quickly show how the kidney data looks like. So as you can see from here, we have like different nodes. We have cortex of organ and kidney and then renal medulla. The center is the body coming to the kidney, and the various loops will be the FTUs, the loop of Henle, the tubules, and then the anatomical structure. These are the anatomical structures. Moving forward, we have like the cell type that is there in the data set, and these are the biomarkers. The green are the biomarkers. As a part of this exploration of the data set we have, we can just compare the data. You can create your own data sheet and compare and just fine tune the data if you want, and then view how each organ is connected with the anatomical structure and the cell type and the biomarker that are linked with it. This was a quick view of the reporter. Coming back to my slides, we have on this page, we have a small of like image of the week where we show tissue organs that we have mapped till now, and it's quite eye-pleasing. It's really good. Yeah, thank you all. And this is if you want any kind of if are there any questions, you can get back to me, or you can just ask away. Yep.
Thank you so much, Supriya. That was a fantastic presentation and a great way to round out this online symposium.
Yeah,
as with all the talks, we're likely going to have lots of questions come in from the audience, and I can see that we've received some already, so I'll move straight onto the Q&A now. Your first question is, what is the integrity of the data that is used for the project?
So as I mentioned before, we are collaborating with multiple institutes, and as a part of this collaboration, we have like subject matter expertise, who really work on the real tissue data. And once we have gained the data from the literature or from like different sources, we accumulate those data and we go to the subject matter expertise to validate the data so that we get the exact information and we are on point about the information that we are going to put it on the portal. Yeah.
Thank you so much, Supriya. Moving on specifically from that question, actually, who validates the data in this project?
So we have like different subject matter expertise for example, in the case of kidney, we generally go with to KPMP team, where they have already mapped and they know how kidney as a kidney organ looks like. So, and as a part of our project, we have a biologist called Ellen Quedicus. She also give us information about the integrity of the data. Is it proper, or is it like, do we have a multiple mapping of the cell type that is there? Are the ontology IDs perfect? Because the ontology database is really huge and we have multiple entries for it, and in which way is it beneficial to our project. So yeah, so that is it. Like we go to subject matter expertise. As a bridge, we speak to our biologist who can help us with the detailing of the information.
Thank you so much, Supriya. We still have time for a few more questions today. So how big is the project and which teams are associated with it?
So the project is for HubMap. We have like multiple team. We have like the registration unit team. It's a team of like 3 members, And then we have VR gallery team, which is a team of like 5 members. And we have a machine learning team, which collaborates with different tissue units to get the cell segmentation of tissues. And then we have we have like a 3D organ gallery, 3D organ visualization also. So we have like we are a team of like 5 to 6 teams with multiple people in it. Yeah.
That's excellent. And we have time for one final question today, which is, what is the technology used throughout this project?
So we are focusing, we are not confined to only single technology. As a part of putting up things into data portal, we have like developers who work on like JavaScript and TypeScript and the different kind of technologies. Coming to visualization, we have people who are working as machine learning people who put up the things into beautiful visually highlighted tissue organs, tissue types. And then we have like for the 2D illustrations, we have medical illustrator. And for like 3D organ illustrations, we have 3D model, like a person who models the 3D architecture. So so we are not confined to single technology, in a short, I would say.
Thank you so much, Supriya. Unfortunately, that is all that we have time for today.
Okay.
Any questions that we didn't have time to get to will be sent to the speaker after the event. And so that's it for the Spatial Biology Revolution 2023. As mentioned throughout the day, all of the talks from today's event will be made available to view on demand for the next 30 days. You can access the talks via the same link you used to access them today, and we will be sure to pass on any further questions you submit through the system to the relevant speaker. That just leaves me to say thank you once again, Supriya, and to everyone in the audience, and we will see you again at our next online symposium, the Laboratory of the Future 2024. Thank you and goodbye.
Thank you, everyone.